hardware devices that sequence.
That's right.
I thought that that's good. Sorry.
Okay, so first, thanks for coming. Today I will talk about some novel computational methods for sequencing data analysis. Here, the sequencing means genomic sequencing or transcript. Or the transcriptome sequencing. As most of you are from computer science background, I would like to start with the central dogma. So from the central dogma, we can tell how the proteins are created from the DNA genes. So in the first step, the genes in the DNA will be transcribed into the pre-mRNAs. So in each gene, there will be multiple exons and introns. And the exons are the coding regions, and they are separated by the introns. So in the first step, both the exon and the intron will be retained, and the pre-mRNA will be created. And in the next step of splicing, the introns between the exons will be removed, and all the exons will be connected together to an mRNA transcript. In the last step, the mRNA transcript will be translated into proteins. So as we can see, the mRNA transcript actually serves as a bridge from the DNA genes to the proteins.
So is this describing what happens in nature or in the computer?
In nature.
Thank you.
So as we know, the DNA stores our genotype information, and most of our phenotype are determined by the proteins. So studying the mRNA transcript, or studying the whole set of all the mRNA transcript, we also call it mRNA transcriptome, can help us to reveal the linkage between the genotype information and the phenotype information.
Can you explain for us genotype and phenotype for those of us who never took biology?
So genotype basically we can consider it as genetic information. So consists of ATCGs, just like the combination of GTAGs will determine which type of protein can be created through the translation, the transcription and translation. The phenotype information basically how we look, like the color, the eye colors, like the hair colors, and all things we can see from in the real world we can see.
This mRNA transcript is a sequence of this whatever is of this thing that are in DNA. Is it unique in the following sense that if you have that one, the third line, you can create back the first one? Or is it not? Or is it can be created by more than one?
So basically DNA consists of the combination of lots of ATGCs, right?
Yes.
So a unique combination of ATGC will create a unique combination of the mRNA transcripts because they're transcribed from this ATGCs into DNA.
So my question was different. Is it a one-to-one fund in the sense that if you have mRNA transcript that was uniquely created by, there are not two DNAs that you create? Or is it just, is it one?
The answer is no, because of the enzymes that are lost.
Really? So, but maybe introns also are uniquely determined by Rafi previously you said that you are not
I didn't say I am not taking biology.
out the function of cells, right? So different organisms, like different tissue in your body, actually carries different sets of proteins because they carry out different functionality. Your eyes have cells for vision, and kidney have cells for other function, right? So the difference of proteins from the same DNA actually come from the difference in the mRNA transcripts. So if you look at it, the pre-mRNA have these exons, right? However, exons are like nodes in the graph. But how are the nodes going to eventually be connected to form a path could vary, right? You can skip exons, and that's
You can have A, B, and E,
yeah.
and then create another transcript.
To have different mRNA transcript from the same gene, and they might vary due to the different cell types. Therefore, generate different transcript, which will be translated into different proteins. So, of course, besides this grafting, which is called alternative splicing, there will be also allelic difference, which the change in TC, ACGT, right? That would also cause difference in protein. Like the mutation in cancer could change the functionality of a protein just by mutate a particular base. So yeah, this whole thing is called central dogma of biology. From DNA to mRNA to protein. And this is bigger. And this entire study here has been on mRNA transcripts. So we want to study the variety of mRNA transcripts to see what are the things specific to the disease, because mRNA transcripts are much easier to measure or to sequence than the proteins. So we study the mRNA transcript for its variety and quantity through the technology called RNA-seq. And here, Xinan's work is on how do you interpret the RNA-seq, which really is the very basic form in terms of raw data out of the sequence machine. So does that help? Yes.
Thank you. So as Dr. said, the RNA-seq is a technology that can help us to profile the mRNA transcript, the whole set of mRNA the mRNA transcriptome. So for each run of the RNA-seq, it will generate tens to hundreds of millions of sequencing reads. So basically, each read can be considered as a subsequence of one mRNA transcript. So when we are when we perform some sequence match task, like to do the read alignment to map the RNA-seq sequencing reads back to the DNA, the reference genome, we can tell where are the exons and where are the introns. That's because the exons are basically the regions covered by the sequencing reads, and the introns are the reads that are the regions that the reads will jump over. Like for this brown, the reads in brown, we can see some of the one part of the reads is in the exon A, and another part is in exon B. So that means this read jumps over the first intron. So with this splice, we call this jump over as splice junction. So from this splice junction, we can infer there is an intronic region here. So based on all those alignments of the reads, we can tell the structure of this gene and how they constitute the mRNA transcript. Besides the structure information, we can also tell the expression levels of the gene. we can simply counting how many reads falling into each exonic region of one gene or one transcript. Then it can be used to estimate the expression level of this gene or transcript. And we can even go further by comparing the estimated expression levels of genes and transcript. And we can conduct some comparison between two groups of samples, like normal and tumor samples. Then we can get a list of differential expressed genes between two different conditions, like normal or tumor. So here is a very typical ANSIG data analysis pipeline. So I listed 4 most common applications here: the transcript reconstruction, transcript quantification, alternative splicing, and differential gene expression. And as we can see, for all those applications, the first and a very fundamental step is the alignment. Is how to align the reads back to the reference genome. So most of my the first of method I will talk about today is about the aligner. So I would like to first briefly go through the history of RNA-seq read aligners. So actually, before the 2009, there is no specially designed aligners for RNA-seq reads. At that time, people used some very general purpose tools like BLAST and BLAT to perform the read alignment. But the performance is very limited because of the algorithm and just because they are not specially designed for this purpose. And between the year 2009 and 2012, the first set of RNA-seq read aligners called TopHat, GSNAP, and MapSpice were introduced in this area to perform fast read alignment, and they focus on the read from 36 base pairs to 76 base pairs because these read lengths are the most common read lengths at that time. And after 2013, the two aligners star and HiSet were introduced. They can perform super fast alignment, and also they can handle the reads up to 150 base pairs. But as the sequencing technology evolves, now some platforms like the Illumina MySeq machines, they can generate reads up to 300 base pairs. And most aligners cannot handle reads with this long length. That's because you can imagine a read, if you have a longer read, it's more likely that that read will span more exons, will contain more splice junctions. So it will introduce some additional challenges for the current aligners. So that's one motivation we design our new aligner. Another limitation for the current aligners are most of the tools only focus on the regular transcript read alignment. But besides the regular transcript, we also have circular RNAs and gene fusion transcript. In the regular transcript, the reads will follow the same order as in the reference genome. Just to say, the upstream of a read sequence will be mapped ahead of its downstream read sequence part, just like these 3 reads. But in a circular RNA, the end of a downstream exon is connected to the start of an upstream exon. So it will create a circular RNA. Then if you map the reads back to the reference genome, you will find out some reads will have the downstream part of the read sequence will be mapped ahead of the upstream read sequence. Another thing is called fusion transcript. In fusion transcript, two separate genes are fused together to create a new MI transcript. So if you have a read crossing this fusion site, when you map this reads back to the reference genome, you will find the different parts of the read sequence will be mapped far apart in the reference genome. So because of those limitations of the current aligners, we developed our new aligner called MapSize 3. So the goal is to meet the following 3 needs. the alignment of longer reads, and the systematic and the unified solution to align reads from different types of transcripts. And also, we want to achieve this the same scale speed of the current fastest tools. So I would like to go through the MAP-3 algorithm with a pair of sequencing reads. So the read sequences is usually from, the length of the read sequence is always from 36 to 300 base pairs long, and the reference genome, for the human reference genome, is about 3 billion base pairs long. So basically the task, the aligner is trying to find a sequence match between this read sequence and this reference genome. Our algorithm is based on the suffix array index of the reference genome. So the algorithm will start from the first base and looks for the maximal mappable prefix between this read sequence and the reference genome. And it will stop when it meets a mismatch between this read sequence and the reference genome. Then it will jump over this base, then restart a new search until it finds a mapping position of the last base of the read sequence. So after this step, we can see the read sequences are naturally segmented into different parts. Some of the segments, like the first one, only has one mapping position, and some segments will have multiple mapping positions. And some short segments, like this one, will have many candidate mapping positions. And in this step, we'll reject the mapping positions for this short segment, just because if we want to, we do not want to sacrifice a lot of speed, like in terms of the mapping performance. But I will introduce you some strategies we developed to resolve this short segment mapping. In the next step, the segments will be clustered based on their mapping positions on the reference genome, and the gaps between each two neighbor segments will be filled, and the gap could be a mismatch, or insertion, or a deletion, or even a splice junction. So here, each candidate cluster can be considered as a candidate alignment for the whole read sequence. As we can see, most of our read sequence have been mapped after this step, but still, there could be some short segments stay unmapped, like this one. Now I will talk about two strategies we developed to resolve this short segment mapping. First one is we can learn from the other alignments, the other reads. For example, for this short segment, we can find out there are many read alignments that cross this same splice junction just next to this read sequence. Then we can easily tell there is a splice junction here. Then we can directly align our short segment to the other end of the splice junction, then get a read alignment of this short segment. Another strategy is called local index. The local index, as I mentioned, our algorithm is based on the suffix array index of reference genome. Besides the suffix array index of the whole genome, we also build the suffix array index for each small piece of reference genome. So in this step, we will only use this local index to support the search of this short segment in this local region. Because this local index is only built for this local region, it's very likely we can only find one unique mapping positions compared to so many mapping positions using the global index.
So, when you talk about context aware, then it basically means your knowledge of your neighbors. Do you have a concept of a neighborhood size, like in natural language processing, when we're looking at phrases of words, and you think about if you do a moving neighborhood window or a moving window?
Right. So usually one sequencing data set contains tens to hundreds million of reads. So we do not align each read one time. So when we get this unmapped segment, we can find a lot of other reads that's mapped to the same region or the neighbor region, right? So that's something we can learn from. So then after mapping this short segment, we can get a complete read alignment of this whole read sequence. So if we have only one cluster that can cover the whole read sequence, that potentially be a regular transcript read alignment or a circular read alignment. If we need 2 or more clusters to cover read sequence, that means it is from a June Fusion transcript. Here I will show some performance comparison we conducted between MapSize 3 and the other tools. The first one is regular read alignment yield. So we simulated data, we simulated reads which lengths from 100 to 200 to 300, and each data set contains 20 million pairs of sequencing reads, and the height of the bar shows how many the number of reads mapped, the number of mapped reads reported by each tool, by each alignment here. So as we can see, for each data set, the first bar is ground truth, and the second bar is MapSize 3. If we compare each tool's result with the ground truth, we can see MapSize 3 always reports the most consistent yield and the distribution of different types of reads compared to the ground truth. The different type of reads is based on how many numbers, the number of splice junctions contained in each read sequence. If the blue ones means this read sequence has no splice junction, and the green ones means there is only one splice junction. The red one means there are more than one splice junctions. And in circular detection performance comparison, we use the real data from a paper described a tool called Siri. So in this paper, the authors validated 24 circular eyes. So for the 24 circular eyes, I plotted the number of reads reported by each tool. So we compare the MapSize 3 with Siri and Star Fusion. So as we can see, almost in all the circular eyes, except for this one, the MapSize 3 reported the most supporting reads for all those circular eyes. And also in June Fusion, we use simulated data of 50 simulated June Fusions. And for each June Fusion, we also plotted the supporting reads reported by each tool. And the white circle represents the ground truth, and the yellow triangle represents the MapSize 3 result. And as we can see, for all those June Fusions, the MapSize 3 reports the most consistent supporting reads compared to the ground truth.
Question. I don't understand why you deal with pairs of reads. Because you didn't use a pair anywhere in the algorithm, did you?
I should mention this actually in the sequencing technology, because for the so there are two strategies of sequencing technology. One of them is called single-end sequencing. Another is called pair-end sequencing. So the sequencing machine will first basically chop the MI transcript into small fragments. And the single-end sequencing strategy will only read the sequence from one end. And the pair-end technology will read the sequence, will read the fragment from both ends. So if we mapped correctly, the two ends of one pair of sequencing reads should be mapped to almost the same region. So that's something we can make use of, because we want to consider all the sequencing, both ends of the pair of the read sequence as a whole to improve our accuracy of the read alignment.
So each pair then is guaranteed to be
from the same fragment.
Same fragment.
Yes.
Okay.
So in conclusion, MapSci3 is very robust to the reads of different lengths, and it's also very comprehensive. Besides the regular read alignment, we can also detect the circular lines and gene fusions. Now we know the MapSci3 can be used to detect gene fusions. But when we get a novel gene fusion, we might ask a question whether this gene fusion is widely present or very specific to a certain individual, cell, tissue, organ, or cell type. Very fortunately, we now have access to a lot of public data sets like TCGA, ICGC, SRA, and we can analyze the data deposited in this database to tell whether this detected novel gene fusion is very specific to your sample or widely present in the population. For example, if we find a gene fusion called FGFR3 to TACC3, there are the two genes in human. So basically, the two genes are fused together in some lung cancer samples. So when we detect this kind of gene fusion, we may ask a question about whether this gene fusion is also present in other lung cancer samples or also in other cancer type samples. One way to do is we can download the data from all the public data sets and perform the tools like MapSci3 to analyze one by one. But that's not practical at all because in our experiment, it takes a couple of hours to process each data set. So if you consider a database called TCGA, we have more than 1,000 sample, more than 1,000 data sets, even for only lung cancer. So another way is to do some search, do some query. Like we can take the gene fusion specific sequence. We can take the sequence, span the two fusion sites here, and because this sequence should be very specific to this gene fusion, then we can query it against all the data sets in this database. But there is no available tools to perform such large-scale query. SQL Server, our solution is SQL Server. Probably some of you may have heard about SQL Server 2 weeks ago in Yes Talk, but I could talk more about the biological aspect, right? So basically, for each data set, it contains tens to hundreds of millions sequencing reads. And all those reads, and this data set can be represented by a list of k-mers. Each k-mer is basically a subsequence of read with length k. So we can, for each data set, we can get a list of k-mers and their frequencies in this read sequencing data. And for a sequence we want to query, we can also get a list of k-mers. Then we can perform some match between all those k-mers against this k-mer list, right? So if we find all those k-mers in this data set, then we can be pretty sure about the presence of this gene fusion in this data set. But if we cannot find any k-mer in this data set, so it means very likely this gene fusion is absent in this data set. So that's for only for one data set. How about for how we scale it to thousands of samples and billions of k-mers? So basically, we need to develop a method to facilitate the fast access to this huge occurrence map to support a super fast query. And in this occurrence map, for each k-mer, we have its presence information for each data set. 0 means it's absent in this data set, and 1 means it's present in this data set. And so our solution is using the hashing structure developed by Ye, still 0, to support our query. But before that, we first need to partition the large occurrence map into multiple submaps. based on their frequencies. And for each occurrence subnet, we use L0 to build a mapping between these k-mers to its presence information in all those data sets. And we also use L0 to build a mapping between these k-mers to its corresponding occurrence subnet. I wouldn't go into details.
Is there anything unique or any important information to be gained by what the string starts with? It starts with G, starts with A, starts with Z. Is there any useful information to be gained?
I don't think so.
That just partitions into 4 subclasses.
But the reason why we're partitioning it into based on the frequencies, so because we want to separate the common k-mers and unique k-mers between different among all those samples.
Very
So basically the L0 can support the classification of NPs into the L sets, and it's very efficient in both in the query and the index construction, and we have and here is an example of how we use SQL 0 to query the gene fusion sequence. For example, if we have this sequence, we can first enumerate all those k-mers in this sequence, and for each sequence we use this. The first level of SQL 0 is an L0 for like to obtain the subnet ID of this k-mer, and then SQL 0 will assign this k-mer into its corresponding subnet. Then at the SQL 0 level 2, within each subnet, the SQL 0 the L0 will obtain the occurrence information in all those data sets based on the indexing structure of this L0. So then we will get a table like this. It basically represents the presence information of all those k-mers in this query sequence. And then we can do some further analysis, like we can set some thresholds to say only the sequences only when the sequences have more than like 90% of k-mers present in some sample, we can say this sequence or this gene fusion query can be found in this data set.
Do you determine that empirically, that threshold value?
So in our experiment, so actually in most cases we require almost only when all the k-mers are in this can be found.
100%.
Yeah, 100%. Yeah.
Okay, not really a threshold.
So in our experiment, we queried around 10,000 fusion sequences over 10,000 TCGA tumor data sets, and we confirmed 92% previously annotated gene fusions in one publication, and also we discovered around 270 novel gene fusion occurrence in all those data sets, and it only takes 5 minutes to return the result, and it's orders of magnitude faster than the existing algorithms. And we have an estimate, even using the most efficient algorithm currently available, it may require around 785 days of computation to get the result of the gene fusion presence. And it only requires 12 gigabytes memory, and it also achieves a great compression ratio. It only uses 76.6 gigabytes to support the query of the sequencing data, the raw sequence data of 5 to 4 terabytes. So in conclusion, we developed a pipeline to interpret and search ANSYS data. It consists of 2 parts. The first one is called MapSci3. It can be used to analyze a single data set and to return the result of like the circular eye detection, gene fusion detection, and also report the alignment for each read sequence. And once we get some novel things, we may want to confirm its presence in the other data set. Then we can use SQL Server to perform a super fast query over large-scale ANSI data sets. So it also achieves a really great compression. That means even for a large data set like large database like TCGA, we only need we can use a very normal desktop to perform the query against all those data sets in the TCGA database. So some references, and I would like to say my advisor, Dr. Xinan Liu, and some collaborators in my lab and also in the College of Agriculture, and also Dr. Chen in UCSC. Okay, I would like to take more questions.
Yes, those are novel, no one else has. However, what I don't know, and I understand that you're comparing against other people's techniques, which I get, but if you all are looking at 54 terabytes of data, how do you know, how are you coming up with ground truth or gold standards or something against which you're comparing so that you can definitively say, oh, my algorithm found these things, and I know that because these poor people sat over here for what should have been 785 days of computation, and they're placing these and comparing these manually. Do you see what I'm saying?
Yeah.
Just the cynic in me says,
Well, so we actually conducted some experiments. We have found like 270